Hello students, welcome to Ligase Academy, your educational catalyst. In this video, let us take a look into some special chromosomes called as giant chromosomes. Just like its name itself, giant chromosomes are those chromosomes which are very large in their size compared to the normal somatic chromosomes. So, they are also called as mega chromosomes. They are generally very long and thick. The major two types of the very familiarly known giant chromosomes are polytene chromosomes and Lamb-Brush chromosomes. Let's move to the first type of chromosomes, that is polytene chromosomes. Polytene means many strands or many threads. So, polytene chromosomes are the large chromosomes with thousands of DNA strands, and they are specially found in the salivary glands of dipterans. They are also observed in the rectal epithelium, trachea, gut, etc. of dipterans. These chromosomes were first reported by Balbiani in 1881, but they were named by Kohler. These chromosomes are many times larger than the normal chromosomes, so that they can be approximately 200 micrometer in length, and we can even visualize them under a compound microscope. So how are these chromosomes formed? We know that during cell division, the chromatin will duplicate during the S phase, and the chromatids get separated by the formation of spindles and the nuclear membrane formations. The cell will divide into two. That is, two sister chromatids will be formed and two daughter cells will be produced. We all know about the cell division. In the case of polytene chromosomes, this is not happening. There is a process called as endomitosis, where the chromatin duplicates but the chromatids do not separate. So there is no spindle formation and no nuclear membrane rupture. So what happens? All the sister chromatids forms will get joined together in that cell itself. Here is an image of polytene chromosome visualized under the compound microscope. So here we can see there are different bands. Some are dark and some are lighter bands, which we will be discussing later. Polytene chromosomes contain alternate dark and light bands. The light bands are also called as interbands. The dark bands represents the heterochromatin and the light bands represents the euchromatin. About 3/4 of the DNA is present in the bands, whereas a few amount of DNA and a large amount of RNA is located in the interbands, which are generally the transcriptionally active regions. At some specific sites, the bands of polytene chromosomes swell or uncoil themselves, which are called as puffs. These are the active region of gene transcription. The banding pattern is usually same in all insect tissues, but the puffs, it differs. The puffing pattern usually changes in developmental stages. And sometimes there are exceptionally large puffs, for example in Chromosomes, and these exceptionally large puffs are called as Balbiani rings. So the puffs are the region where the RNA synthesis takes place. Coming to the structure of polytene chromosomes, there are mainly 6 arms in a polytene chromosome which are arising from a chromocenter. So what is a chromocenter? We know that there are chromomeres in every chromosomes, and all the chromomeres of the chromosomes are centrally fused together, which we call as the chromocenter. And from the chromocenter there are 6 arms, 5 long arms and 1 short arm. Among the arms, the X chromosome arm is the longest one, and The fourth chromosome arm is the shortest one. Here, in case of male flies, we know that there are Y chromosomes, but a Y chromosome is not visualized here because it is usually seen as fused together with the chromocenter. Looking at this image, we can easily visualize the light and the dark bands and also the puffs. The puffs are usually occurring because of the uncoiling of the DNA. So the major significance of these types of polytene chromosomes is in the study of evolutionary changes at chromosome level. Also, it helps in the study of temporal control of gene expression because we know that the bands can be recognized by their thickness because it is different in different bands and spacing is different, so it will help in different types of studies. Now let's move to the second category of giant chromosomes, that is Lamb-Brush chromosomes. These are first observed by Fleming in 1882 in the salamander oocytes, but they are named and described by Ruckert in 1892. The structural interpretation was carried out by Gall and Callan. So these are large-sized chromosomes which are usually seen in the diplotin stage of meiosis in the yolk-rich oocytes of many vertebrates like fishes, amphibians, etc., and in some other small invertebrates also. Regarding the size, in invertebrates, the Lamb-Brush chromosomes are smaller compared to the vertebral Lamb-Brush chromosomes. And also it is present in a single cell algae called as acetabularia. Bird and Bottineau in 1989 used the term test tube brush chromosomes for Lamb-Brush chromosomes because they look similar to the structure of test tube brushes, so they were called as test tube brush chromosomes. The Lamb-Brush chromosomes are usually present in bivalent conditions. So it consists of a main axis with 4 chromatids of the 2 bivalent chromosomes. So that each Lamb-Brush chromosome contains a central axis region where the 2 chromatids are highly condensed. And each chromosome has several chromomeres which are located throughout the length of the axis. And from these chromomeres, the chromatin loops out to form the lateral loops. Another unique feature of Lamb-Brush chromosome is their very unusual extended meiosis. The diplotin stage is very prolonged and it may take several hundred days to several years. In amphibian oocytes, the early development occurs very rapidly because the independent locomotion and feeding is attained as soon as possible. So, this will require the production of materials for early development even before fertilization. In such species, the yolk is accumulated and the genes for producing mRNA are amplified. The chromatin of these chromosomes give out fine loops at the lateral sides and thus it will be looking like a Lamb-Brush. The loops are always symmetrical but the centromeres of the chromosomes bear no loops. There will be about 10,000 loops per chromosome. About 10% of the DNA is present in the loops and the size of the loop usually increases with the genomic size. Each loop is covered by a matrix that consists of RNA and associated proteins. Thus, each loop of the Lamb-Brush chromosome is found to perform intense transcription of the RNA molecules. The loops are very much significant because they are used in the observation of transcriptional changes, hybridization analysis, and in chromosome mapping because every loop appears at a particular point in the chromosome. That's all about giant chromosomes. Thank you for watching the video. Stay tuned to our new videos on cellular organization. Please watch, like, share and subscribe Ligis Academy, your educational catalyst.